thank you for the opportunity to present here. I would to thank the seminar series. Here, I will be presenting my work from Xavier Trepat Lab from IBEC Barcelona. I work at Columbia now, but I just started. So today I will be presenting epithelial shells. So like epithelial tissues are usually like a robust tissues playing key role in many processes like compartmentalization of organs and morphogenesis. And for us, the most significant part of it was that in vivo, most of the epithelia is curved and folded into 3D structures. It can be simple curved as like a early embryo, or it can be highly folded and branched as lungs. So even though it exhibit range of geometries, a common epithelial organization is that of a cell monolayer enveloping a cavity. And as you can see here in these examples, and I'm showing you all these static images and from the previous talk, you know that it's dynamic, and epithelial reshaping is a physical process. Like cells have to move, stretch, do things to make these shapes happen. And physical forces such as pressure, tension play a key role in driving and regulating these changes. A good example is a cyst, so which is a pressurized tissue with uniform tension, and the shape it produces is a uniform curvature, a sphere. But if you have a tissue, pressurized tissue with elongated tension, like a oriented tension, it produces a elongated geometry such as tubes. On top of this, everything is dynamic and multi-scale. So there is a long-range cell movement, and there is nanoscale molecular activity. So considering all of these aspects, we ask a big question, like what physical principle guide the epithelial shape? And we thought about answering this question by attempting to build the structure in vitro so that we could control shape, pressure, tension, and also understand the material properties of the tissue. So for this, we basically had two requirements. One was control over geometry and physical forces, and the second one was make direct mechanical measurements. So considering the geometry of lumen and cavity, we thought epithelial shell could be a good way of starting. It's a sphere. Let's try to make a sphere. It feels like a simple geometry. But we realized that it's quite difficult to satisfy both of these requirements with a spherical geometry because it's closed. So we thought, like, instead of doing spheres, we do spherical cap. So the way we went about this was to invent a microfluidic chip. It basically has two channels separated by a porous membrane, and we coat this porous membrane with a fibronectin pattern, which is our adhesion promoting protein, in this fashion where you have circular region of non-adhesion. And we coat our porous membrane with MDCK monolayer. So MDCK cell line is a very widely used epithelial cell line, comes from canine kidney cells. And our assumption was when we apply hydrostatic pressure from below, it could, like the cells at the lower adhesion point would detach and they would inflate into a pressurized geometry, a spherical cap. And we will have pressure, we will have tension, and with the confocal imaging, we will be able to see it. So does it work? Luckily, it works. And here in this video, you can see this is top view and this is side view, that we are able to dynamically control the pressure and image the shape with confocal imaging. Here you can see in detail that the cells are able to stretch 3 to 4 times their original area, and all the while it maintains a spherical cap geometry, which is a good idea for us because it makes our life easier to measure radius of curvature. And with just pressure and radius of curvature using Laplace law, we can calculate tissue tension. So this allows us to try to characterize the material property of epithelia with measurement of tensions and strain. When we were trying to image this in confocal microscopy, it takes a minute. So it's a big structure. It's a 100 micron tall structure. For us to image that much, it takes more than a minute. And what we realized that the dynamics of these structures were much faster than we could image. So to still able to get tension strain values and characterize the mechanics, we decided to take advantage of the symmetry of the system and image only a single line of pixels in 3D. So you can see that we can see the tissue here. It's a bit low quality, but it's good enough for us to measure tension and strain. And what we saw that the tissue inflates and moves, stretches fast to reach a steady state shape and tension. So what we saw that the plateau tensions were around 5 to 10 millinewton per meters. So this is a typical range for active tensions produced by actomyosin cortex. So here we started thinking that we should probe the mechanics at the actomyosin timescale. And for this, we subjected these shells to different rates of triangular pressure waves, one as 20 seconds and another 2000 seconds. So for faster rates, we saw that the tissue was stretching and stretching more and more for every cycle. So pressure goes from 0 to 200 Pascal and comes back to 0. But strain, you see that it ratchets up till it reaches a limit cycle. So it oscillates between two. And then for slower cycles, we saw something opposite where cells were able to stretch to really high strains and then come back down to 0 at the end of each cycle. So there was large reversible deformations. Taken together, all of these experimental findings, we thought that it's clear that there is a rate-dependent response. So to understand this, this whole project has been a very close collaboration between Professor Marino Arroyo group with Adam Usseri and Pradeep Bal. So what they consider, and in line with literature on epithelial monolayers, that the actomyosin cortex plays a big role, as you see in the previous talk as well. So what they have done, they have created a model which models cells as collection of actomyosin cortex. So where every cell surface is an active gel. So I will not be able to go into details, but the model accounts for three aspects. First, viscoelasticity. So basically you have actin network responding to deformations by reorganizing the network and turning over filaments. Plus it also accounts for contractility, which is responsible for active tension. This is typically due to activity of myosin. So the way we understand this is that there is a total tension when we inflate a flat tissue to a curved geometry. The total tension in the tissue can be done in two components. One is a viscoelastic component and another is an active tension. So when tissue is stretched for a very short time scale, we call it viscoelastic time scale, it can store viscoelastic tension. But the moment you stretch it, the active actomyosin filaments are reorganizing themselves, turning over, and this dissipates that viscoelastic tension, so that at larger time scale, the only tension which remains is the active tension, which is due to myosin activity. So this can be understood in our simulations of cyclic stretching. So if we stretch tissue faster than viscoelastic time scale, you have this ratcheting response, because cells can stretch, but they don't have enough time to unstretch, and the next cycle begins. So this is how the strain accumulates. However, if we deform them very slowly, so the cells have enough time to dissipate viscoelastic stresses and stretch to larger deformations and come back to original, because there is enough time for cells to deform. So with this, we understood how the tissue mechanics work for stretching and intention. So we thought, what about negative tensions? So it's a perfect talk to follow. So compression plays a big role, particularly in morphogenetic events, in case of hydra, in case of tissue folding. So as we have this nice system where we could create shells with positive tension, we started to think, can we do negative tensions? And Laplace law came to our help. So Laplace law is basically product of pressure and radius of curvature. So we thought if we could just flip the sign of this pressure and make it negative, we could have negative tension. So we decided to try this out in simulations where we deflated the shells which are in steady state to minus 100 Pascal. But we did it two ways, one slow and one faster than viscoelastic timescale. So what we saw, if we deflate a tissue faster than slower than viscoelastic timescale, so the tissue deflates by maintaining spherical geometry. It always is spherical cap and it becomes flat. And this is because the viscoelastic tensions are being dissipated as we are deflating the tissue. And so the active tension remains dominant and the total tension never goes below zero. So we never introduce compressive stresses in the tissue. So if we do it faster than viscoelastic timescale, there's not enough time for viscoelastic tension to be dissipated. So the total tension will reach zero and negative. So what happens here that the cells don't have enough time to shrink their area. So what they do, they produce accumulation. And this is what we refer to as buckling. So any like a thin shell you could have, like when you compress it, it buckles. And this is what is happening, but instead of a thin sheet, we have a spherical sheet. So with the model, we understand there are two important parameters. One is deflation time and another is active tension, which controls the buckling. So deflation time is kind of obvious that if we deflate something very slowly, like in this case, the shell will always maintain spherical curvature because there is enough time for cells to dissipate viscoelastic tensions and it comes down flat. But if we do it faster, there would not be enough time, tensions will go negative and the tissue would buckle. So a nice uniform curvature becomes kinked and there is folds appearing here and here. So this is the hallmark of buckling, that symmetry breaking event. So we decided to probe the active tension component of this. So the idea is that the more active tension, the shell would not buckle. And what we saw by adding calcicolin, which increases myosin activity, the case where tissue would have buckled does not buckle. And the other way around, when we tried in a slow case where tissue does not buckle, where we reduce active tension by adding blebistatin, we see that the tissue does buckle. So we understood the parameters of controlling buckling and material properties, just active tension and deflation. So the next question we asked, what is the outcome of this buckling? So buckling is typically associated with buckling patterns, buckles, wrinkles, creases. In many natural systems, buckling and compressive stresses have been implicated in various pattern formation at all the scales, like from subcellular scale to organ level scales. And funnily, I recently found the same compressive stresses are also responsible in driving creases in pumpkins and raisins. So for us, what we decided to look at was to try to see at different size what is the effect of deflation, like fast deflation and buckling. So here we are looking at shells from top at the base plane, and we saw there are two different kinds of folds emerge as a post-buckling pattern. In this one, you can clearly see that the shell comes down in the middle first and creates accumulation around the periphery. But in the larger size terms, larger size shells, we have radial patterns. So the question is like why there are these two different patterns. So what breaks this symmetry of like a peripheral fold to radial fold. So we went back to the model, and this is where Pradeep was working on this. And so our model is very computationally expansive. So we can do few cells, but for a shell size of a millimeter, it's too big. So Pradeep simplified the model into a continuum model. So where we lose the detail of cells, but capture all the physics. And when he was doing the simulations, he reproduced the peripheral fold. But problem was that in all the cases, like all the size shells, they were buckling the same way, where it would produce a peripheral fold. So we were a little stuck. And this is where we looked into literature and found a single author paper from 1950s from a little company called Shell. And so that this actually pointed out that our shells are not just buckling in vacuum, they are buckling in a viscous medium. So there is a length scale of folding is associated with the viscous medium. So you can imagine that when tissue deforms, it just does not deform itself. It also has to deform the liquid around it. So it has to move the medium up and down. And the viscosity plays a big role for this. So when we ran simulations of increasing medium viscosity, we found that it is indeed an important scale which drives the length scale. So if the viscosity is low, the tissue can make a one large big deformation, as you can see in this dimple. But if the viscosity is too high, it is difficult for tissue to make large deformation because you will have to push large volumes of media around. So what tissue does, a compromise where it deforms a smaller level at multiple bases. So here you can see the length scale affected here. Just by increasing medium viscosity, the length scale goes down. This is the length scale for the dimple, and the dimples get shorter and shorter. And this is what produces radial folds. So with this information, we plug back the model with the viscosity included, and what we saw was what we were getting in the experiments, that the smaller shells were producing peripheral fold, but larger were forming more complicated internal folds. And this you can appreciate well if I show you the length scale. The length scale remains the same. It's the same length scale, but different size. And if the size of the shell is comparable to the size of the length scale, then the only choice it has to form a peripheral fold. So with this, the same logic we thought was also applicable to actin cortex. So just to think about it, the actin cells are basically, for us in our model and in our mind, is just a bag of actomyosin cortex. And cortex is very thin, it's 100 nanometers. And in this situation, you have compression acting on a cell, and cortex is also getting compressed. It is surrounded by two different mediums. You have cell culture medium on the outside. Inside the cortex, you have cytosol, which is more viscous than cell culture medium. So by this logic and the length scale, we could predict that at subcellular scale, you could have 1 micron size folds. So to visualize this with Thomas Wilson, we got a device, which is a light sheet device, where we could image the same experiment, but in light sheet. And we had much better spatial and temporal resolution. So all of these events are happening at second scale. So for us, it was a feat to get this data, but it was also very expensive in terms of size of the data. But we could monitor shape of the cells. Here you see a nice steady state shell, which is very spherical in geometry. And we could see details of cells. And when we look at individual cells, we see that the apical surface and basal surfaces are flat, like of uniform curvature at least. And for junctions, they are straight. And here is a segmentation of one cell. But when this shell undergoes buckling, we catch one frame of buckling in light sheet microscope. We can see that the uniform curvature is no longer there. And individual cells, each surface, apical, basal, and junction, are buckled at a scale we had predicted of 1 micron. So that was pretty satisfying. We actually confirmed that what we are seeing is because of actin cytoskeleton. So by imaging membrane with the F-actin live stain, what we saw that the membrane and cortex follows exactly the same path, which indicates that actin is involved in buckling. Plus, when we saw that how long does these folds last, we saw that they last only for the time scale of actin turnover. So it becomes wrinkle geometry becomes flat. So it's not good to remember that the flat geometries are because of the active tension. So like the myosin contractility produces a flat geometry, and it creates a tension which creates cells to be like foam or bubbles. Uh, for us, these things are transitory. Like the subcellular scale folds are transient, but for larger tissue scale folds, which we saw earlier, were lasting. So the next question and final question we asked was, how do we rationally guide these folds? So in spherical geometry, the curvature is uniform. And because of symmetry, curvature is uniform and the buckling pattern is difficult to predict. So we could not tell which way folds form. So we imagined a uneven curvature surface. Imagine a donut. So you have like one direction, there is another curvature, and in other axis, there is another curvature. So you have two different curvatures in two different directions. So when we deflate this geometry, the two different curvatures would deflate at different rates. So this is highlighted in this example where we have inflated a triangular shape and you have non-uniform curvature in different directions, the curvature is different. So when it deflates, it creates a pinching form where along the medial axis. So we expect that the fold would be along the medial axis. This we tried it in different shapes, ellipse, triangle, and rectangle. What we found that indeed along the medial axis folds were able to form. So ellipse would form a fold along the middle, a triangle would form a fold along the Y, and rectangle would form a fold along double Y. So we reproduced this in our experiments, and we satisfyingly we saw the same result. So you see that here, just by controlling the footprint of a shell, we could form a very unique geometries of tubes, which are very difficult to do. So if I tell you, like, get me a Y in a tissue, it's not possible. But it opened up the engineering potential of this system just by a few parameters. So to conclude, I hope I have presented a very nice system where we could control shape and pressure in epithelial tissues. We explored mechanics of the shells at actomyosin timescales. Plus, I think we discover a very unique physics of these materials, which are hierarchical material, where they are able to buckle at tissue scale as well as subcellular scale. And there is a possibility of engineering tubes. So to conclude, I would like to acknowledge the funding sources and my labs, like Trepat Lab, Arroyo Lab, Rokagusak Lab, and also Nerurkar Lab. I especially want to thank Thomas Wilson, Tom Golda, Pradeep Bal, Adam Ozeri, and Pao Pujol for all their help. I thank you all for attending and giving me the opportunity to speak. One last thing on internet, like this community, we are building a community called Epithelial Mechanics Fan Club. So please find us and reach out if you want to contribute. Yamini did it. So thank you.
Okay, perfect. Great job, Nimesh. Okay, let's go to questions. The first one is from Dibyo Majumdar. Dibyo, would you like to ask it yourself? Okay, I'll ask it for them. Um, is this phenomenon tissue or cell type specific? Will difference in molecular motors make a difference? Nimesh.
Uh, so I think actomyosin network is everywhere and it plays a big role. I think as we've seen in Hydra, in many tissues, like it's one of the key players. And the buckling also is like quite well established now that in many systems you have seen buckling at tissue level, but at subcellular level, not so much. But there are few examples in nature, if you see Drosophila, like there are during certain stages you develop curly junctions, and these curly junctions are also at the same length scale as our subcellular folds. So, but the only problem is that in many contexts in nature we have matrix around the cells. So it's quite difficult to probe the mechanics of epithelia. So in our study, we are able to do that just isolating the tissue and trying to apply compressive stresses to it. I hope that answers the question.
Yamini, you had a question?
Yeah, hi. It's the first time I see this. So I had a question based on tweaking the epithelia. So because you just added myosin rigidity, but can you do like what happens when you do blebistatin? What happens? Can you do multiple monolayers instead of one? Like, can you get a giant thicker thing and then have and then map? Because it's not usually one monolayer, no, that buckles, at least in vivo. So I'm just curious for perspective.
So yeah, it was a fantastic question. So we actually did the blebistatin, and so in our conception that the active tension is driven only by myosin. So we were looking at the ways we can affect myosin. So we found that the calcicolin could increase myosin activity and blebistatin could decrease myosin activity. So, and the results we got were like as expected. But in case of like multi-layer tissue, we could do it. We have not done it in this study, but I think it's a very interesting idea to try to work with it. The only issue is with so here you see, because it's a monolayer, we have an advantage of like it has low bending rigidity, so it does not have to bend so much. It can easily bend, stretch. But if it's thicker, there would be it would be more difficult for it to bend. So we have not tried that, but could be interesting place to start. Like I think would be a good idea. Try.
The next person who wants to ask a question is Minkush.
Yeah.
Minkush, are you there?
Thanks, Nimesh, for the very nice talk. Actually, had a question about the numerical simulations that you have.
Wondering, when you have these shells and you have them of different footprints, do you
Uh,
have cell shape gradients or cell area gradients on the dome?
It's a very good question. We have looked at it. That so in this slide you can see. So we saw that the cells were stretching uniformly, so there was not so much heterogeneity. So cells on the top were stretching as much as on the side. There was like some cells which would stretch much more, but there was not something localized that only cells on top stretch more than side. If so, there is a previous study from our lab where they show that there is a heterogeneity in cell strains, but that happens when you are stretching cells very, very slowly, like longer than actin time scales. So here we were focused only on actomyosin time scale, which is like less than 1,000 seconds. But if you stretch tissues over hours, you could see the heterogeneities.
Another quick question I wanted to ask, and when you alter the footprint, you still keep so the density of cells in every case is the same, right? So your
Yes.
footprint alters the number of cells that are participating in this dome.
Yes.
Okay.
Yes, you're right. So this is actually the key. I could not go into detail here, but the idea is to change the thickness. So in buckling phenomena, thickness matters. So if your structure is very thin, it would have a different phenotype of buckling than a very thick structure. So in our experiments, what we saw that the thickness of the cells was the same. So here you have three different sizes,
Mhm.
but all of them have the same thickness. It's just the size is increasing from 100 microns to a millimeter. So relatively, the 1 millimeter structure is much thinner than 100 micron structure. And this thin, thickness of the tissue, relative thickness of the tissue actually plays a big role in the length scale of folding. Yeah, this is one of the also reason that it buckles the way it buckles.
Great. Thanks very much.
Thank you.
Sorry, Minkush, you got cut off. Could you?
Yes. Actually, I'm just wondering because I'm just starting to model some viral capsids, where is like the spherical cap, actually spherical shapes, and they have these different defects of pentamer so that it can be folded into a sphere. And there these so I'm trying to also study the buckling of that. And where I was reading a couple of papers actually just today where the buckling also depends on the defects and the size of the shell. But I guess yours is very like continuum shape and depends on the thickness and all. So I was just curious, how do you model it and what's the numerical technique, like finite element, or what exactly are you using there?
So very good question. Unfortunately, like I did not do the modeling. So our collaborator, like Marino Arroyo Lab, did it. So it's a vertex model. So it's a finite element simulation, but the idea is that in epithelia you have like a lot of these models, like vertex model. So where you model each cell as a hexagon, but in what they have done, they have extended it to a new level where you have each cell surface with a mesh which can deform. So that's what made it very expensive. So what we did, we simplified the model by adopting this new geometry which Pradeep came up with, where it is modeled as a continuum, a bilayer. So, like the viral cascade is a fantastic model because it does buckle and it does form the shapes. So, like what we see in our simulations of like these dimples and the length scales, the viral cascades does do that. But what is very difficult to do in experiments is that they are very sensitive. Like these buckling patterns are also very sensitive to defects of like thickness. So if you have like a tiny defect in the thickness, it would trigger buckling at that region. That's why we could only predict it qualitatively, that the peripheral folds are becoming radial folds. But we could not predict exactly where the fold will occur because the material is heterogeneous with different material properties and thickness. But yeah, it's very exciting that you're working on viral cascades.
Okay. Yeah, thanks. I will probably get in touch at some point, but thank you. Very nice talk.
The next question is from Meghna. What makes the epithelial sheets remember the cap shape? Not sure if remember is the right word. Under expansion and compression.
So under tension, like under inflation, it would remain spherical because of the active tension you have. So you imagine like a bubble. So when you inflate a bubble, it always remains spherical because there is uniform surface tension. So what we see in our tissue that when we pressurize it, it behaves something like a bubble. So it has a uniform tension, even if the cells have different properties or thicknesses. But what is unique about this system is that when you pressurize it, it takes a surface tension geometry like a sphere. So it always is spherical cap. But when we put compression, it does not need to be spherical cap because now there is no positive surface tension to make it spherical cap. So it buckles as you see here on the slide.
I also had a couple of questions to Nimesh if time allows. So it was beautiful that you were able to model this process with very few parameters, and in the model actually you can see that the shapes are so when the when whatever blows up, so the whole epithelium blows up, right? And then when it's buckling, it's actually reproducibly forming the same shapes in the model. So I was wondering if in the experiments you were able to like get these shapes in a very reproducible manner, or if it was a bit like deviating.
So we got it like qualitatively similar patterns. So we could clearly see that there is a peripheral accumulation, there is internal folds. But what we tried to do was like, we had this idea of like we could catch the amount of radial folds there would be. But it was very difficult to do that in experiments. We could predict that there would be radial fold or peripheral fold, but we would not be able to match exactly. So this is why we changed the shapes. So instead of like doing a spherical shape, we tried to do like break the symmetry in different directions. And then we were able to produce reproducible folds. For spherical, the only thing we could do is peripheral or not peripheral. We could not predict the orientation. It was difficult to do like this. Yeah.
And just curious, so do you have any ideas on what could be the effect of osmolarity during this dome, like when it's under tension and when it is buckling? Or have you thought about this? Just curious.
No, we have not thought about this properly because for us, so with like osmotic shocks, you could create like shells. But for us in our system, we are able to create them directly with the pressure, like hydrostatic pressure. So we did not probe osmolarity at all. But what we know that these shells are not uh, like as watertight as I thought they would be. So there is leakages. But at the time scales we are dealing with, those don't play a role because we are working at a very fast time scales. Like all the buckling events are happening under 10 seconds. So everything happens within 10 seconds. So there is no, I would not attribute any role to that. But we have not investigated that. I think it's a good question.
Yeah, thank you very much. Yeah, very cool talks.
Thank you.